Hello, my name is Eric Bushong, and I work at UC San Diego in the laboratory of Mark Ellisman, the National Center for Microscopy and Imaging Research. At NIGMR, we've been performing volume EM since 2009, starting with 3D serial block-based imaging systems, and nowadays we also perform quite a bit of array tomography of thin sections on silicon wafers. And we use X-ray microscopy a lot in our volume EM workflow, and I'll talk about later how we find XRM useful for doing volume EM. So at NIGMR, we look at a lot of different organisms and tissue systems, and even occasionally something interesting like mud. But we spend a lot of time looking at mouse brains. And so I was asked to talk in today's session about how we prepare mouse brain specimens to optimize ultrastructural preservation and staining for volume EM imaging. So here's a mouse brain specimen, specifically the cerebellum, that was prepared at NIGMR and stained with serial block-based imaging. And in this volume, you can see that other than a little bit of charging in the nucleus of this Purkinje cell here, the bulk of the specimen has no charging artifacts, even though it's being imaged at high vacuum, and that's because it's been rendered conductive through the staining process. All of the cellular processes seem to be full of cytoplasm. None of them are extracted. If you zoom in on the cytoplasm of this Purkinje neuron, you can see there's a ton of details here. In particular, the membranes are intact and smooth, high contrast. The mitochondria all look very happy and have beautiful cristae. You can clearly see cytoskeletal structures, macromolecular complexes, nuclear pores. There's just a ton of stuff and details in this specimen due to the way that it was preserved and stained, and they can all be captured in block-based imaging. So this is what I would call a gold standard for a well-preserved, well-stained mouse brain. So to begin with, if this talk is supposed to be about how to optimize the ultrastructure of a mouse brain for EM, I should probably spend about the next 10 minutes talking about how to perform transcardiac perfusion with aldehydes, because it really is the critical key step for getting good ultrastructure. And of course, perfusing a brain for electron microscopy is not the same as for LM. And I just want to quickly point out a few of the important details that we pay attention to when we do our perfusions. First of all, obviously, you would need to pay attention to the osmolarity and pH of your solutions themselves to make sure they're all correct and proper. The perfusion pressure should be high enough to make sure that blood is quickly washed out of the brain and replaced with fixative, but not so high that you're popping blood vessels. You want to make sure that your flushing solution is oxygenated and heparinized to help with the flushing process. And as for the flushing itself, there's only a couple of milliliters of blood in a mouse, so there's no point in flushing for very long. You want to get the fixative to the brain as soon as possible. And then we add calcium because divalent cations in your buffer help to stabilize membranes, and it's been shown since a long time ago that it avoids holes that can develop between adjacent plasma membranes in the brain. And finally, since aldehydes don't actually fix membranes themselves, we begin to chill the brain as soon as possible to stabilize and reduce the fluidity of the membranes. So even during the perfusion, we're chilling the brain, and by the time you take the brain out of the skull, it should already be cold, and it's kept cold at least through the first osmium step, because that's when you first start actually fixing the membranes themselves. And as for post-fixing the brain, we always make sure we give it a strong post-fix with glutaraldehyde in the refrigerator, because osmium tetroxide is a pretty harsh fixative actually. It cuts proteins and it can extract the tissue. So a good strong fixation with aldehydes helps a lot to prevent that. And then moving on, another step of the specimen preparation protocol that can strongly influence, especially the membranes and ultrastructure of membranes, is the dehydration process prior to embedding the tissue in epoxy resin. This is best performed as cold as possible and even at sub-zero temperatures, preferably. Most labs, including our lab most of the time, do this process on ice, but it has been shown that a progressive lowering of temperature protocol can be adapted for preparing fly brain for Fixation imaging, as was reported in this paper by Xu et al. And I'd like to briefly discuss a protocol that we recently published that shows that you can use high pressure freezing and free substitution of brain tissue for volume EM imaging. So typical free substitution protocols don't really stain tissue heavily enough to allow for imaging at high vacuum, and they also don't preserve genetic probes such as Apex2 or Minisol. So the protocol that we developed with the Chi and Su lab at UCSD, which we call CryoChem, allows you to do both. Here you see slice of mouse brain that was 100 microns thick. It was fixed with aldehydes for a primary fixation, and then high pressure frozen and free substituted, and stained and imaged by serial block-based imaging at a high vacuum. And then you can see throughout this volume, there are axons. If I back up a little bit here, we can see axons that are labeled with Apex 2 label. Here's one right here that's very nice. So this tissue generally has very nice preservation. The myelin sheaths are intact. There's no extraction of the cytoplasm. The membranes are all smooth and round, as you expect for typical free substitution material. And the synapses are very easy to detect throughout the tissue. So I'm going to move on from the fixation of the tissue to staining the tissue with heavy metals for block-based imaging. Well, actually, before I talk about that, I should say that the staining protocol that you use is going to be pretty heavily influenced by the imaging methodology you're using. You have block-based imaging, where you're going to put a largish block of tissue in the microscope and image the block whole, and you need to make sure that this whole block of tissue was sufficiently stained to render it conductive and provide sufficient signal and contrast during imaging. And then you have other volume EM approaches where you first cut a series of thin sections from your specimen and collect them on some substrate. And so for these approaches, you can, of course, do a lot of heavy metal staining on the sections, the epoxy sections, and you don't need to achieve this heavy staining of the whole block of tissue. And then you have hybrid approaches, for example, the gas cluster ion beam SEM method that was reported by Hayworth et al. And they cut thicker sections and collect them and then do imaging on them. And of course, there's other considerations and factors on determining what your section thickness should be. I just want to quickly point out that you can stain some pretty thick epoxy sections up to a few microns thick with on-grid staining or on-section staining with heavy metals, as we've been doing for many years for ultra-high voltage electron tomography. So when doing block-based imaging, where the tissue needs to be stained on block prior to imaging, we developed a protocol several years ago that's typically called ROTO, stands for reduced osmium dialcarbohydrazide osmium. And it's generally used by a lot of the community for doing volume EM imaging in one form or another. There are a lot of variations out there now, but these are the basic steps in the protocol. And it starts with an initial osmium staining step using reduced osmium with potassium ferrocyanide. And this gives much better membrane contrast than using regular osmium tetroxide. And then you lay down a mordant, usually thiocarbahydrazide, to allow a second layer of osmium tetroxide to be stained on top of the first layer. And this renders the tissue really conductive and highly electron scattering. And then you follow up the osmium tetroxide labeling with on-block uranyl acetate labeling and on-block lead labeling. And then finally dehydration and epoxy embedment. So this is the protocol that gives the sort of results that I showed at the beginning of the talk with the cerebellum. The downside, the primary downside of this protocol is that it only allows specimens up to a few hundred microns thick to be stained well throughout their extent. And any deeper, and you start to first of all see these very heavy, spurious deposits of stain, and then a sudden diminution in the staining intensity. So some steps in this protocol have been modified by other labs recently to improve the staining penetration. For example, there's the BROPA protocol that was developed by Mikula and Dink, where they use oromide in their osmium staining step to improve the penetration of osmium tetroxide. And they also just use much longer staining steps than we typically use. And then there's this paper by Hua et al., where they split up the reduced osmium step into first osmium tetroxide and then followed up with potassium ferrocyanide. And this alone was able to significantly improve the penetration of the staining and allow specimens up to a millimeter thick to be stained with the ROTO protocol. I just like to quickly point out, as an aside, I guess, an approach that we take when we have specimens that either cannot be stained very heavily with these stains, or they have a lot of open spaces, open epoxy maybe. And sometimes it just happens where we have specimens that for one reason or another, sometimes the ROTO stain just doesn't stick. It kind of fizzles. And so we put it in the electron microscope and we see charging artifacts. Nowadays, that's never really an issue because we use focal charge compensation. And this is where you put a small nozzle right next to the surface of the block. and you blow a stream of nitrogen onto the block face, and this dissipates charging artifacts. And so this approach, it provides much better image quality than you get from traditional variable pressure SEM. And we've been able to run very long runs, up to 6 months long, using focal CC. And you can image even just blank resin with focal CC without seeing any charging. Now I'd like to turn to how we use X-ray microscopy in our volume EM workflow. And first of all, we use it to screen our specimens before beginning volume EM imaging to make sure they do have sufficient staining and they don't have other defects or artifacts before we invest in a big volume EM run. So here's examples of several 1.2 millimeter thick cores of brain tissue that were stained with variations of the roto staining protocol that I just showed. And it's immediately apparent looking at these images, these micro CT slices, which areas of each specimen were stained well and which areas were not stained well. You see some areas where you have a very heavy deposited stain that's probably not going to look very good in the electron microscope. You have other areas where there's clearly insufficient staining. You can also see small holes and vacuoles that may suggest you had poor perfusion or fixation of the tissue. And you can see defects like cracks in the tissue that you would want to avoid or reject the specimen for those reasons. Unfortunately, with a typical laboratory-based XRM, it's not possible yet to image at sufficient resolution to really assess ultrastructural preservation in these specimens. Generally, with XRMs, you have a CCD sensor you're using to collect the projection images. And so for one thing, the bigger the field of view you try to acquire with a large specimen, that means you have to have bigger pixel sizes and lower resolution. And then also just the optics of most laboratory-based XRMs are not going to allow you to collect much better than a little less than 1 micron spatial resolution. But still, Nakula and Dink did show in their report on the Broca protocol that if you can stain a whole mouse brain, they were able to image it in a Zeiss Versa XRM, which is a laboratory-based XRM platform. And then they were at least able to assess at low resolution the staining quality and uniformity throughout the whole brain. And I'd just like to very quickly point to a very recent paper that came out by the Lee Lab at Harvard University, where they used a synchrotron in Europe to perform X-ray holographic nanotomography. And here they were able to get pixel resolution down to 20 nanometers. And at this point, it really is possible to potentially assess ultrastructure in your specimen before moving on to electron microscopy. The way that we use X-ray microscopy in our lab is, first of all, to assess our specimens before moving on to volume EM, but also to precisely target regions of interest so that we can limit our volume EM to just the areas we care about, and also be able to correlate the volume EM data sets with other imaging modalities like confocal imaging and 2-photon imaging in vivo. So here's an example of a brain where cells expressing GFP were first imaged with confocal microscopy, and then a specific cell was targeted, and that cell was found in the X-ray microscope, and that guided the targeted acquisition of a serial block-based volume of that cell. The EM volume was registered with the confocal data using a nuclear stain, DRAC5, and this stains the nuclei and the heterochromatin, which is visible in all 3 imaging modalities, the light microscope, the X-ray microscope, and the EM microscope. And so the registration we achieve allows us to precisely register cell bodies and major cell processes. And from there, we can segment the EM data down to the finest processes to assign them to a given cell. So finally, before I wrap up, I'd like to point to the largest connectomics projects that I've been involved in. It wasn't mouse brain, but it did take advantage of several of the techniques I discussed in the talk. And that was imaging an entire Leech ganglion, which is about 500 microns by 500 microns and 100 microns thick. This project was done in collaboration with Daniel Wagenaar's lab at Caltech. You can find a manuscript on bioRxiv. So this connectome was particularly interesting because we first imaged the ganglion with voltage-sensitive dyes during stereotypical behaviors of swimming, crawling, and bending. And each neuron's behavior during these activity during these behaviors was characterized. We use this in a microwave enhanced version of the roto staining protocol, which allows us to, we thought, slightly improve the staining throughout the ganglion and get more uniform staining. We use micro CT imaging to screen our specimens and make sure they had no cracks or preservation artifacts and good staining. And then we use the micro CT data to guide the SBEM run to make it as targeted as possible. And so in the end, we were able to pan the voltage sensitive dye imaging to the EM data, reconstruct an entire neuron, the DE3 neuron, and see how the inputs of neurons associated with each of these stereotypical behaviors impinged on the target neuron. So this concludes my talk, and I thank you for listening.